Today I'll be talking about negative inducible control in the lac operon. The lac operon is responsible for breaking down lactose into galactose and glucose. An operon is defined as a regulatory area that controls multiple genes and transcribes them to make proteins needed for the breakdown of lactose. This happens when lactose is present in the cell. If there is no lactose, we do not need to make these proteins because that would waste energy. The lac operon is a negative inducible control, meaning the DNA binding protein is a repressor and prevents transcription when bound. However, when a small molecule known as the effector is present, it induces transcription. Let's think of negative inducible control like driving a car. When the repressor is bound to the DNA, we are pushing the brakes, so transcription is off and the car will not move. When the effector is present, though, the repressor doesn't bind, so transcription is on, kind of like taking our foot off the brake so the car can move. The regulatory gene known as LAC-I codes for the repressor, which will bind to the DNA at the operator known as LAC-O. LAC-P is the promoter, which is where RNA polymerase will bind. The operator region overlaps with the promoter region. This means that if the repressor is bound to the operator, then RNA polymerase cannot bind to the promoter region. The structural genes are LAC-C, LAC-Y, and LAC-A. These three genes code for the proteins as follows: beta-galactosidase, permease, and transacetylase. In this lac operon with the wild-type genotype, every gene has normal function and lactose is absent. The regulator protein is transcribed and translated and is floating around in the cell, and it goes and binds to the operator. When the repressor binds to the operator, it prevents RNA polymerase from binding to the promoter, which means we cannot transcribe our structural genes. There is no lactose in the cell to be digested, and this is why none of the structural genes will be translated and transcribed. This lac operon has the same genotype as the previous one, except lactose is present, so we want to use lactose as an energy source. The lactose molecules are converted into allolactose by beta-galactosidase. Allolactose can bind to the repressor protein, which changes the shape of it, and now the repressor cannot bind to the operator. The repressor cannot bind to the operator, but RNA polymerases can now bind to the promoter and transcribe the enzymes needed to break down lactose within the cell. This mutant genotype is a partial diploid, meaning the cell is diploid for just this one lac operon section. For this example, lactose is absent. In this genotype, we have a superrepressor labeled as IS. The superrepressor cannot be turned off and can bind to both operators. The superrepressor will repress on both pieces of DNA, whether lactose is present or absent. There is also a non-functional repressor and an OC. The superrepressor is going to bind to the operator and turn off transcription. The superrepressor will also try to bind to the OC, but since it is a mutated operator, the DNA sequence is different, so the repressor cannot bind to it. RNA polymerase will then bind to the promoter and transcribe LAC-C and LAC-Y, so we produce functional beta-galactosidase and functional permease still.